Hello students. So in the last class, we were discussing about PCR primer designing. So I'll go directly to the slide that we were discussing. So here you can see this is the locus number AH82464 for which we had designed the primers. Our question was to design primers for full length amplification of the gene and further cloning into the vector PBR322. So in the last class, we learned how to design the primers for amplification of the gene. And accordingly, we had designed the forward primer and the reverse primer. Okay. So now we will further understand how do we select a suitable restriction enzyme for cloning into the vector, and how do we for cloning into the vector, and how do we add the suitable restriction enzyme target site to our primers. Okay. So first thing that we need to do is to look at the vector sequence or the vector map. So as you can see here, the map of PBR322 is visible. And here you can see different unique restriction enzyme target sites of the vector, which are visible in blue, which includes SAL1, PAMH1, ECO-RB, HINDI3, ECO-R1, PST1, and NDE1. So basically, when we look at the sequence of the vector or the map of the vector, we have to select a unique restriction enzyme. Means restriction enzyme that is digesting the plasmid only once. So when we do the digestion, the plasmid should open up or form a linear structure from the circular structure. And similarly, the enzyme that we are choosing must not be digesting your insert internally. Otherwise, that will lead to fragmentation of your insert. So we need to look at the sequence of the gene for which we need to design the primers. So I'll go to the KEC site for this. Again, the gene number is AH82464. So I need to copy this, and I will go to the KEC site to get the sequence. I'll paste the gene number here and hit the search tab, from which I can navigate directly to the gene, page, where if you scroll down, you can reach the nucleotide sequence of the gene. Okay. So we need to copy the gene sequence first and analyze it using another site, that is add gene sequence analyzer, or analyzer. So you need to click on this link, and you can paste the sequence here. Now you can submit the gene sequence. So there are various tabs that are present over here. Okay, and right now we need to look at the digest tab. So we click on digest, and here you can see unique is auto selected. So unique means those restriction enzymes that are digesting this insert sequence only once. Okay, so here what we are interested in rather is to look at the list of non-cutters. Okay, we are looking for those restriction enzymes which are not cutting our sequence, because we do not want that our sequence gets fragmented after PCR amplification when we digest with the restriction enzyme. Okay, so we have to look at the list of non-cutters. So here is my list of non-cutters. So I have to select an enzyme that will digest the plasmid once and will not digest my insert. Okay, so I have the list of enzymes here, which are present in the plasmid. So I'm going to just note them down alphabetically. So I have BAM H1, BAM H1, I have eco RV and eco R1, RV, eco R1, ND3, SAL1, NDE1, and PST1. So these are my options which are present in the plasmid. Now I have to find out which of these are suitable for use for cloning of my insert. So I'm going to select out of this list, I need to select those enzymes which are not digesting my insert. So I go back to the sequence and here is the list of non-cutters of my insert sequence. So I can see BAM H1, ECO R1, ND3. So BAM H1, ECO R1, ND3. I cannot see ECO RV in the list. NDE1. NDE1, SAL1 and PST1 I need to check. SAL1 also I cannot find in the list. And PST1 also I cannot find in the list. If I go back to the list of all cutters, then I can see PST1 digest my insert, SAL1 digest my insert, and EcoRV. These three enzymes are digesting my insert, so they are not suitable for cloning. So the ones that I have highlighted in red, they are not digesting my insert, so they are suitable for cloning purposes. So I can select any one of these. So if I go back here to the map, just paste the list. So these are the ones in red are my option. BAM H1, ECO R1, ND3, and NDE1. Okay, but I would like to go ahead with BAM H1 for the simple reason that it is present in the tetracycline gene. So if I can do cloning here, then the plasmid, the recombinant plasmid with the clone will be tetracycline sensitive because the gene will get disrupted. And it will be ampicillin resistant because the other antibiotic marker is still will be still intact after cloning. Okay, so I'm going to show you how can I insert BAM H1 sequence in the primers. Okay, so for this, I'm just going to copy. Okay, I'll edit in the same slide, BAM H1. So I need the sequence for BAM H1 first. You can just type BAM H1 sequence and then you have the sequence for the BAM H1 enzyme. So you can simply copy this. So I'm also okay, so I'm going to copy it from here. GGATCC in 5' to 3' direction. Okay. So GGATCC in 5' to 3' direction. Okay. So I need to simply copy this sequence and paste it at the 5' end of my primers. Okay. Here and here. Now I'll remove this double symbols. This I need to delete. From here, I need to delete this. Okay. And so after amplification of my gene, they will be, this BAM H1 sequence will be present towards the 5' end of the sequence. Okay. Also, we need to put some 4 random nucleotides above the restriction enzyme sequence, okay, towards the 5' end, because the restriction endonuclease cannot sit at the end of the DNA sequence. So we need to add some random 4 nucleotides before the restriction enzyme target sequence. So these are your primers basically. And we also need to just check their TM and the GC value. So for which we can copy them. And I'll go back to the thermo TM calculator site. Click on the link. And you paste the primer sequence here, first primer or the forward primer and the reverse primer. Okay, so as you can see, the GC content of the first primer is 48.4%, and the second primer is 57.6%. So this is in the required range, which is between 40 to 60%. Coming to the TM values. The TM of the first primer is 72.2 and the TM of the second primer is 72.9. So according to the rule, first of all, the TM difference between the two primers should be less than 5 degrees apart. And secondly, like if 55 degrees is the typical annealing temperature that we are planning to use, then the melting temperature or the TM of the primer should be at least 5 degrees above annealing. So if we, for a 55-degree annealing temperature, the TM needs to be at least 60. So this is well above that. So the primers are fine. So that's it about how do we select a suitable restriction enzyme site for our primers for the cloning purpose. I hope it is clear now. Thank you.